Hello everyone, welcome back to our channel Pharmacy. In this lecture, we will study the concepts of cell division. It will include introduction to cell division, types of cell division, phases of cell division, then interphase and M phase of mitosis, and last will be significance of mitosis. So, let's start. Cell division is a process in which a parent cell divides into two daughter cells. Reproduction of cell takes place by splitting into two and new daughter cells are formed. Daughter cells themselves divide and grow. This is a diagram of cell division in which mitosis and meiosis both occurs. In this part, chromosome number is same as parent cell, that is 2n to 2n, so it's mitosis, and in this chromosome number becomes half, that is 2n to n, so it's meiosis. Types of cell division. There are two types of cell division, that is mitosis and meiosis. Mitosis is also known as equational division or somatic cell division, and it is subdivided into two, interphase and mitotic phase. And second type is meiosis. It is also known as reductional or reproductive cell division, and it is divided into two categories, that is meiosis I and meiosis II. Meiosis is more detailed and complicated as compared to mitosis, so it will be discussed in next lecture. In this lecture, we will study the concepts of mitosis. So, phases of cell division in mitosis are interphase and M phase. Now, we will start with the phases of cell division in mitosis. There are two phases, interphase and mitotic phase. Interphase includes G1 phase, means gap 1 or growth 1 phase, S phase, it denotes synthesis of DNA, G2 phase also means gap 2 or growth 2 phase, and G0 phase, also known as resting phase. M phase has several phases, that is Prophase, Metaphase, Anaphase, Telophase and Cytokinesis. It occurs in the nucleus. It is a simplified flow chart of cell division. Cell division is divided into two parts, interphase and M phase. Interphase is further divided into four categories, that is G1 phase, S phase, G2 phase, and last is G0 phase, which is a resting phase, and cell do not divide in this phase. G1 phase takes 10 hours for preparation of DNA synthesis. S phase takes 9 hours for DNA synthesis, and G2 phase takes 4 hours, in which growth continues and cell is prepared for M phase. Mitotic phase includes prophase, metaphase, anaphase, telophase and cytokinesis that takes only 1 hour. So process of cell division is completed in 24 hours. Now, we will understand each and every phase in detail. So, let's start with G0 phase. In this phase, cell do not divide but metabolically active, and this phase is also known as resting phase. Next is G1 phase. It is also known as gap 1 or growth 1. This is the phase between preparation of DNA synthesis and initiation of DNA synthesis, means this is the period before the synthesis of DNA. Cell is metabolically active and continuously grows. In this, DNA is not replicated. S phase. It is also known as synthesis phase because synthesis of DNA takes place and amount of DNA per cell is doubled but chromosomes numbers remain same. Last phase of interphase is G2 phase. In this, cell growth continues, and this is the phase between end of DNA synthesis and the beginning of prophase. Proteins are also synthesized for preparation of mitosis. Our next topic is mitosis. It is the process by which a cell replicates its chromosomes and then segregate them by producing two identical nuclei in preparation for cell division. This phase takes 1 hour for completion of cell division, and it occurs in the nucleus of cell. It is also known as equational cell division because chromosome number in the parent cell and daughter cell is same as shown in the diagram. First phase of M phase is prophase. In this, initiation of condensation of chromosomal material takes place, means chromosomes become untangled. It begins after interphase. DNA has already been replicated when cell enters prophase. Replicated centrosomes from interphase move apart towards opposite poles of the cell, powered by centrosome-associated motor proteins. And at the end, Golgi bodies, endoplasmic reticulum, nucleolus and nuclear envelope disappears. Next phase is metaphase. In this, complete disintegration of nuclear envelope occurs. Process of condensation of chromosomes is completed. Metaphase chromosomes are made of two sister chromatids and held together by centromere. Kinetochore is the site of attachment of spindle fibers, and chromosomes lie at equator with one chromatid of each chromosomes as shown in the figure. Anaphase, it is the third phase after prophase and metaphase. It separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughters. Metaphase plate split along with the centromere. Chromatids also separates. This phase is characterized by microtubule disintegration. At this stage, the chromosomes are at the maximum level of condensation. This helps in easy segregation and formation of the nucleus. And this phase is characterized by the movement of chromosomes to the opposite poles with the help kinetochore microtubules. The microtubules create a central force by attaching them to the centromere of chromosomes. The attached microtubule depolymerizes and becomes shorter and pulls the chromosomes apart towards the centrosomes. Next phase is telophase. It begins once the replicated, paired chromosomes have separated and pulled to opposite poles of cell. This phase is characterized by breakdown of spindle fibers and nucleolus. During telophase, a nuclear membrane forms around each set of chromosomes to separate the nuclear DNA from the cytoplasm. It accounts for about 2% of the duration of the cell cycle. Golgi bodies and endoplasmic reticulum appears in telophase. Chromosomes begin to uncoil, which makes them diffuse and less compact. In the diagram, nucleus is separated but cytoplasm starts to divide and nuclear envelope reappears. Last phase is cytokinesis. Cyto means cell and kinesis means movement, that is process of division of cytoplasm. It is the physical process of the division of a cell. The cells are divided by the formation of a cleavage furrow on both the sides of mother cell as the cell membrane constricts at the cell equator during cytokinesis. This process cannot occur without karyokinesis. Cytokinesis begins in anaphase and ends in telophase. It ensures that nuclear and cytoplasmic contents are accurately partitioned between two nascent cells. And it often happens after nuclear bonding. Our last topic is significance of mitosis. It is responsible for development of the zygote into an adult. Equal distribution of chromosomes to each daughter cell. It is responsible for growth and development of an individual. And it maintains the purity of the genome as no recombination or crossing over takes place. Also maintains the constant number of chromosomes in all the body cells of an organism. It is responsible for the repair and regeneration of old and damaged cells. For examples, gut epithelium and blood cells. In the next lecture, we will study the simplified concepts of meiosis. Thank you. Keep watching and take care.